Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive.
Yeah.
Today, we're tackling something huge, chronic pain. And, the way we treat it often feels blunt, right?
Definitely. Like a sledgehammer, as you said.
Exactly.
Yeah.
But what if there was a different way? Imagine dialing down pain, not by blocking nerves directly, but by tweaking the fluid around them. A massive new study in Nature has found this gene, SLC45A4, that seems to act like a volume knob for pain. It controls these molecules called polyamines, and that directly affects how sensitive we are to heat and chronic pain. It's pretty wild.
It really is a fascinating angle.
So before we really get into the weeds on SLC45A4, maybe let's just quickly touch on why this is such a big deal. Chronic pain, it's not trivial.
Oh, absolutely not. We're talking about, 1 in 5 adults worldwide. It seriously impacts quality of life. It's a major cause of disability.
Wow, 1 in 5?
Yeah. And honestly, our current treatments, they're often just inadequate. Lots of side effects, not enough relief. So finding new approaches is critical.
And the basic biology, how we feel pain, that comes down to specific nerve cells, right? Nociceptors.
That's right. Nociceptors. They're our body's danger detectors. Specialized neurons that sense anything potentially harmful: heat, pressure, chemicals.
Okay.
And how easily these neurons fire off a signal, their excitability, that's directly linked to how much pain you perceive. In chronic pain, these guys can get way too sensitive.
Firing off when they shouldn't be.
Exactly. Sending pain signals constantly.
So where do these polyamines fit into the picture? What are they?
Right. Polyamines. Things like putrescine, spermidine, spermine. They're these small, positively charged molecules, and they're basically everywhere in our cells. Super common.
Okay.
They do lots of things: cell growth, signaling. But the key thing here is they can interact with ion channels on neurons.
Ion channels, those are like the gates controlling electrical signals.
Precisely. So by interacting with these gates, polyamines can actually tune the excitability of those nociceptors we were just talking about.
Okay.
We knew their levels often change in chronic pain states, but the specific mechanism, the how, was murky.
And that's the gap this study fills. We suspected polyamines were involved, but we didn't know the exact system controlling them in pain pathways or have that direct link to human pain intensity.
Exactly that. We had these pieces, these hints, but not the clear connection. We were missing, the specific transporter, how it manages polyamines in the nervous system, and proof that messing with it actually changes pain sensitivity in people. This study really connects those dots.
Okay, let's unpack this then. How did they even zoom in on SLC45A4? It wasn't exactly a famous gene in pain research before this, was it?
Not at all. This is where the power of big data genetics comes in. They used what are called genome-wide association studies, GWAS.
GWAS, right. Scanning loads of genomes.
Yeah, basically. They looked at the genetic data from the UK Biobank, that's over 132,000 people, and searched for tiny variations in DNA, these SNVs, that correlated with how much chronic pain people reported.
And SLC45A4 just popped out.
It did. Specific genetic markers right near or in the SLC45A4 gene showed a really strong association with higher chronic pain intensity. It wasn't just some random hit, it was a clear signal pointing right at this gene.
And you mentioned this finding was robust. They checked it elsewhere.
Oh yeah, absolute crucial. They didn't just rely on the UK Biobank. They replicated the findings in other massive data sets like the Million Veteran Program in the US and the Finnish Genetics Project.
Wow.
Seeing the same link pop up consistently across these different huge populations, that really tells you it's likely a genuine biological effect.
Makes sense.
And even more interesting, they did a broader analysis of GWAS looking at what else these SLC45A4 variants are linked to. And it turns out it's associated with a whole bunch of other things besides pain.
Like what?
Things like osteoarthritis, certain immune conditions, metabolic traits, even some psychiatric functions. It suggests SLC45A4 is involved in something pretty fundamental, not just a one-trick pony for pain.
Okay, so the human genetics were practically yelling that this gene mattered for pain. But the name SLC45A4, It doesn't exactly roll off the tongue or tell you what it does.
Yeah.
So what's its job?
Right. That was the million-dollar question. And honestly, before this study, SLC45A4 was kind of a mystery protein. There were some old, weak hypotheses linking it to transporting sucrose based on distant relatives, but the evidence wasn't strong. Its real function was unclear.
So how did they crack it? How did they figure out its actual role?
They got clever. It was like molecular detective work. First, they did this broad screening across thousands of different cell lines. They looked for any metabolite, any small molecule whose levels went up or down consistently when SLC45A4 expression changed.
Testing a wide net.
Exactly. That pointed them in the right direction. Then they did direct tests. They took purified SLC45A4 protein and tested if things like sucrose, GABA, or various polyamines would actually bind to it. The polyamines were the clear winners, especially spermidine and spermine. When they added those, it caused a measurable change in the protein's stability, a technique called thermal shift assay, which is a strong sign of direct interaction, like a molecular handshake.
Ah, okay, so it grabs onto polyamines.
Precisely. And then, using cell-based experiments, they nailed it down. They confirmed SLC45A4 is indeed a polyamine transporter located on the cell surface, the plasma membrane.
So it's like a gatekeeper for polyamines moving in and out of the cell.
Exactly. It seems to transport several types: putrescine, spermidine, spermine, maybe with a slight preference for the smaller ones, and it works best at the normal pH range you find in the body.
It's pretty elegant, moving from that huge genetic signal down to the specific job of one protein.
It really is. They didn't just find the gene, they figured out its function, gave it a clear role: polyamine transporter.
But they didn't stop there, right? You mentioned they looked at its structure. See how it works.
That's right. They used cryo-electron microscopy, cryo-EM, which is this amazing technique that lets you visualize molecules in incredible detail. They solved the 3D structure of human SLC45A4.
What does it look like?
Well, it has a fairly common structure for transporters of its class, the major facilitator superfamily, or MFS fold. Imagine 12 corkscrews, these helices, embedded in the cell membrane, forming two bundles with a channel down the middle. It was caught in an inward open state, like it was ready to release something inside the cell.
Okay, standard transporter stuff so far.
Mostly, but here's the really unique part. It has this large chunk, a plug domain, sticking out inside the cell cytoplasm, and this plug actually extends into the central cavity of the transporter itself.
A plug, like it blocks itself?
Kind of, yeah. It seems to act as an autoinhibitor, like a built-in brake. Specific parts of this plug interact with the polyamine binding site inside the transporter, almost mimicking how a polyamine would sit there.
So it regulates its own activity. That's clever.
Very clever. The structure gives us clues about how it controls the flow of polyamines, not just transporting them, but gating that transport. Seeing the structure helps visualize that physical gate.
Okay, so we've got the gene linked to human pain. We know it transports polyamines. We've seen its 3D blueprint with this cool plug feature. Now the big question. What does this all mean for actually feeling pain? Where is this transporter working, and what happens if it's missing?
Right, connecting it back to sensation. First, location. The researchers looked in mice and found that the message for making SLC45A4, the mRNA, is really highly concentrated in the dorsal root ganglia, the DRGs.
And DRGs are central stations for sensory information, including pain.
Exactly. These are the neurons whose endings are out in your skin and tissues, detecting stimuli, and whose cell bodies are clustered in the DRGs near the spinal cord. They're the first step in the pain pathway.
So it's right where the action is.
Precisely. And importantly, it wasn't just in one type of sensory neuron. It's expressed in pretty much all the major subtypes in the DRG. Different kinds of nociceptors that detect heat, mechanical pressure, chemicals, and neurons that detect normal touch.
So it could potentially influence lots of sensations.
Potentially, yes, although its expression levels seemed a bit higher in the larger neurons, which often handle touch and proprioception. But it's definitely present in the pain-sensing ones, too. And when they put a tagged version of human SLC45A4 into these mouse DLG neurons, where did it go? Right to the plasma membrane, the cell surface.
Perfectly positioned to be that gatekeeper.
Couldn't be better positioned. Its location really reinforces its potential role as that volume knob for modulating sensory input right at the source.
Okay, so what happens when you take that knob away?
Yeah.
They made knockout mice.
Right.
Mice without the SLC45A4 gene. You mentioned something about hair color.
Yeah, that was an interesting side observation. The knockout mice had this temporary salt and pepper coat color when they were young. which sounds random, but actually, polyamines are known to be involved in making melanin, the pigment in hair and skin.
Ah, okay, so that fits.
It does. But the more critical findings came from looking at their metabolites. When they measured the levels of different molecules, it was clear that losing SLC45A4 really messed up polyamine balance.
How so?
Well, for example, spermidine levels dropped in the spinal cord, but putrescine levels went up in the DRGs themselves, and spermidine was actually higher in the blood plasma. So the whole system of polyamine regulation was definitely out of whack.
But the neurons themselves looked okay structurally.
Yeah, that's important. They checked the basic anatomy, like the density of nerve fibers in the skin, and it all looked normal. So the wiring wasn't physically damaged, it was the tuning, the chemical environment that was changed.
Okay, so the polyamine levels are off. Did these mice actually feel pain differently?
That was the key behavioral question. And yes, they did. Their basic movement was fine, they explored normally, but their pain responses were altered.
How? Less pain. More pain.
Less sensitivity specifically to heat. When they put them on a warm surface, like a 48 or 50 degrees Celsius hot plate, the knockout mice took significantly longer to lift their paws.
Interesting. So they tolerated heat better.
Exactly. And in another test, a thermal gradient, they actually preferred to spend time in warmer zones compared to normal mice. It suggests their whole perception or coding of heat was shifted.
Wow. Any other pain changes?
Yes, they also showed reduced pain behavior in the first phase of the formalin test. That test involves an injection that causes immediate sharp pain followed by inflammation. The reduction was in that initial acute pain phase.
But not all pain was affected.
Correct. And this is really important. Their responses to mechanical stimuli like light touch or a pinprick seemed totally normal, and their response to noxious cold was also unchanged.
Okay, that is remarkable. It's not just turning down all sensation. It's selective for heat in that specific type of tonic pain.
Precisely. It suggests SLC45A4 isn't a blunt instrument. It's involved in tuning specific pain modalities. Imagine having a drug that could just target, say, burning pain without affecting touch sensitivity. That's the kind of specificity this points towards.
That would be revolutionary compared to current painkillers.
Absolutely. Oh, and one related finding in the knockout mice were also better on a rotarod test, which measures motor endurance. And that correlated with lower levels of the neurotransmitter GABA, specifically in the part of the spinal cord controlling movement where SLC45A4 is also expressed. So its effects might extend beyond just sensation.
Fascinating. So we see the behavioral effects, less heat sensitivity. How does SLC45A4 achieve this at the cellular level? What's the actual mechanism changing the neurons firing?
Right, getting down to the nitty-gritty. They used patch clamp electrophysiology to record the electrical activity of individual DRG neurons.
Listening in on the nerve cells.
Basically, yes. And they found something very specific. There was a selective reduction in the excitability of one particular class of nociceptors, the IB4 negative, non-peptidergic ones. These are often described as polymodal nociceptors, meaning they can respond to multiple types of stimuli, like heat and mechanical pressure. In the knockout mice, these specific neurons fired less readily when stimulated with electrical current.
So less excitable, specifically that type.
Exactly. And they confirmed this using a different technique called ex vivo skin nerve recording. They took a piece of skin with the nerve still attached and recorded signals directly from the nerve fibers while stimulating the skin.
What did that show?
It showed marked hypoexcitability, less firing, specifically in the C-fiber mechanoheat nociceptors, CMHs, which are essentially those C-polymodal neurons again. They responded less strongly to mechanical pokes and even more dramatically less strongly to heat stimuli.
But other nerve types were okay?
Yes,
Yeah.
other types of nociceptors and the mechanoreceptors that sense normal touch were firing just fine. The effect was really focused on these C-polymodal neurons.
Why them specifically? Any ideas?
Well, the likely reason is that those particular neurons happen to express higher levels of certain ion channels that are known to be sensitive to polyamines. Channels like TRPV1, which is famous for detecting heat and capsaicin, or maybe TRPV3 or TRPV4. So if SLC45A4 is controlling the local polyamine concentration around these neurons, and these neurons have polyamine-sensitive channels, they become the ones most affected when SLC45A4 is gone.
Okay, it's all coming together. The genetics pointed to the gene, biochemistry showed it's a polyamine transporter, localization put it on sensory neurons, and the knockout studies show losing it disrupts polyamines, reduces heat pain behavior, and makes those specific C-polymodal nociceptors less excitable.
You've got it. SLC45A4 acts like a fine-tuner for polyamine levels, and that, in turn, dials down the excitability of key heat-sensing pain neurons, a metabolic dimmer switch for specific pain types.
This really feels like more than just discovering another pain gene. It feels like a whole new way to think about pain control.
I agree. It's a significant conceptual shift. A lot of previous pain genetics focused, quite understandably, on the ion channels themselves, like NaV 1.7 mutations causing pain disorders, or on neuroimmune interactions.
Right, directly targeting the signaling machinery or inflammation.
Exactly. But this study puts the spotlight on a transporter that controls the metabolite environment around the neuron. It's not about directly blocking a channel, it's about modulating the concentration of these natural molecules, the polyamines, that tune the channel's activity.
Metabogating, as they call it.
Ah.
Modulating the gates via metabolism.
Precisely. And that metabogating strategy might offer a way to develop analgesics that avoid some of the really serious problems we see with opioids, or even some channel blockers like addiction or complete numbness.
Plus, the way they connected all the dots from the huge human GWAS data all the way down to the molecular structure and the specific neuron excitability. That's quite a feat.
Absolutely. That GWAS to function pipeline they used, integrating human genetics, biochemistry, structural biology, electrophysiology, animal behavior, it sets a really high standard for validating genetic findings and understanding the underlying biology. It's very thorough.
So where does this leave us? What are the next steps? This sounds incredibly promising for future treatments.
The potential is definitely there. The immediate and probably most exciting next step is medicinal chemistry. Can we design small molecules, potential drugs, that specifically target SLC45A4?
To either block it or boost it.
Exactly. Inhibitors or activators. If variants linked to less pain correspond to, say, less SLC45A4 function, then an inhibitor might mimic that protective effect. That's the hope.
Okay.
Then we need more detailed mapping. Where exactly is SLC45A4 expressed in human DRG neurons? Knowing the precise subtypes will be crucial for therapy development.
Right, translating the mouse findings to humans.
And, of course, testing any potential drug candidates. They'll need to be tested rigorously in preclinical models of different types of chronic pain, neuropathic pain, inflammatory pain, to see if they actually work and are safe.
And could this idea, this metabogating, apply elsewhere, beyond pain?
That's a really exciting possibility. Could similar mechanisms, transporters controlling local metabolite levels that tune neuronal excitability, be at play in other conditions? Maybe chronic itch, which shares some pathways with pain, or migraine, or even conditions affecting mood or cognition. It opens up a new way of looking for therapeutic targets.
It really is a whole new frontier.
Yeah.
Moving beyond just blocking signals to subtly adjusting the environment that shapes those signals. It could genuinely change how we approach treating chronic pain.
That's a potential, yes. A novel mechanism brings novel therapeutic possibilities.
So if we can find and control this volume knob for pain by targeting a transporter like SLC45A4, it really makes you wonder, doesn't it? What other hidden metabolic controls, these subtle rheostats, are operating in our nervous system, tuning our mood, our memory, maybe even just how we perceive the world around us?
It's a fascinating thought. How much of our experience is shaped by these quiet background metabolic adjustments?
There could be a whole layer of regulation we're only just starting to uncover. Lots more deep dives ahead, I think.